So hi everyone, it's great pleasure to invite Professor Erez Braun in Technion to the sophomore summer school. Actually, this is second invitation. We invited him to the school since 2019, 2 years ago. Today, he will give a talk about morphogenesis. Let's welcome Professor Erez Braun, please.
Okay. Thank you very much. Do you hear me?
Yeah, perfect.
So, yeah, unfortunately, it's in Zoom, not in beautiful Korea. So it's a good afternoon for Korea, good morning for me in Israel. So I would like to thank the organizers for the invitation to this school and the opportunity to talk here. I just want to mention one thing is that if you have urgent question, please jump in at any moment. I cannot answer chats and stuff during the talk, but please jump in and we'll stop and discuss. I think it's more important to get the message and the ideas clear rather than covering more and more material. I will try to avoid the jargon, and there's no requirement for pre-knowledge of biology or anything. The emphasis is actually on the physics, but of course it's on living systems, so there will be all kinds of things. So if I do it wrong and I do mention jargon or something that is not clear, again, please jump in and stop me. The subject I would like to discuss is the physics of morphogenesis, a subject that we work for the last few good years on, but it's part of a larger program of research for me to try to understand physics of living systems from different directions. Then eventually, hopefully, we will be able to build a framework for understanding living system as part of physics. And so the emphasis for morphogenesis, we are explaining in a second what it means. The end message is that from the physics point of view, we might want to regard morphogenesis as a dynamic phase transition. So, and this is not ignoring the biology or anything like that. There are a lot of morphogenesis, as you see, it's in the forefront of biological research. It's one of the important subjects in biology, but the physics approach should go in parallel, and actually the questions and the approaches and the tools are a little bit different. So what is morphogenesis? It's the emergence of functional patterns in living systems. So this is morphogenesis in the context of development, because morphogenesis is used for also to describe other pattern forming system. And in development, this is a pattern formation process, but it's a special type of pattern formation system, because it has to do with the emergence of functional patterns. And this is something that is not very familiar in physics, where we demand that the pattern will be functional. So in this example, you see the cheek development, and along a developmental trajectory, the form is emerging, but this form is a living creature with eyes, with the right symmetry, with legs, with head, all the parts in place. And there are two specific or two general, I would say not specific, two general questions that at the basis for what we would like to understand about this process. One question is, how does the specific shape emerges? And in specific shape, I don't necessarily mean how the cheek details are developed, but how the cheek becomes a cheek and not something else. And I'll show you in a second that this is highly non-trivial. And of course, in biology, the escape is always into evolution and to say that, okay, there was a program developed specifically for the cheek, specifically for human, etc., etc. But I will try to give you a picture that deviates strongly from this approach. And the second question, which directly touches what I'm going to discuss, is what type of dynamics ensure the robustness of the process. So you see all these developmental processes, and we all know, are bombarded by fluctuations, by changing environments, by all kinds of interruptions, and nevertheless they robustly go on. Otherwise, we would not have this talk here, because if it wouldn't be such a robust process, then defects would take over and life would become impossible, at least life for multicellular organisms. And this is a general question I'm also interested in other contexts, is that I regard the living system as a class of dynamical systems. And what type of dynamical systems these are, it's an open question, you know, and it's very central to my research. So this is the outline. Let me go quickly over it. So I'll first determine morphogenesis in animal development. I want to say a few general things about the nature of the process, which highlight also type of interesting things that emerge when starting to think about development. I'll then introduce Hydra, regeneration, as a model system for morphogenesis and explain why this is the spherical curve for physicists. Actually, an organism and a model system that allows manipulations and research that is prevented in many other organism, but is allowed by Hydra, and I'll explain why Hydra regeneration actually and development actually complement each other in morphogenesis. Then I'll come to our research, and I would like to discuss two things. One is the role of mechanics in morphogenesis, and this will do through the description of actin fiber organization in Hydra regeneration. But then the central part of this talk would be the application of electric fields on Hydra regeneration, showing two interesting directions. So I'll separate them. One is reversal of morphogenesis under electric field. So I'll show that morphogenesis in Hydra regeneration is a reversible process. And actually there is a critical point where you can drive the system forward and backward around this critical point. And then I'll go on after this and show that we can study fluctuation and correlations, since we can halt regeneration at a point by applying external electric field. We can start to study fluctuations and correlations at the edge of regeneration, which is an approach similar to what we'd like to do in phase transitions. And then I'll summarize. Okay, so animal development, it's a long story, right? It is a long history, but it can be summarized very nicely by this type of figures. So this is the fruit fly, Drosophila embryo, which is a syncytium, so it's a continuous media at this stage. And pretty early in the fly development, a pattern emerges. The colors that you see here are molecules, concentration of molecules. These are not just molecules, these are proteins that are essential for development. And you see that early, very early, this is a sequence in time, very early in development, there is a well-defined pattern that emerges, and the system ceases to be uniform, and this pattern later emerges as the body plane of the fly. Okay, and so the emphasis here is on chemical or biochemical processes, genes, proteins, etc. And this this was discovered long time ago, but then, of course, it's not unique to the fly, although the fruit fly is a little bit special in the way this pattern is developed, but patterning in protein expression appear also in all other species. This is an example from the zebrafish, and this materialized later as an adult animal. Okay, so this is the basic picture. But then one of the advances, actually one of the great discoveries in the 20th century was that developmental processes are universal. What you see on the top here are a bunch of embryos, different embryos. One of them is human. There is fish and mouse also. And at this stage of the embryo, the shape of the embryo is almost identical. You wouldn't guess which one of them is human unless you saw this picture before. And the point was that it's not only that the shape is very similar, is that all the molecular mechanisms that that have to do with development also are shared between all species. Actually, most of the molecular mechanisms are shared from worms to fly to human, the same molecules and very similar molecular mechanisms play a role in development. So these guys start at some point like this, but eventually they develop more specific shapes. So this is human, and you see fish here, etc., chicken, etc. So this is not unique only to vertebrates that you see, or chordates that have a notch chord, but the shape is unique to this type of vertebrates, but the molecular mechanism, as I said, is completely universal. This was a great discovery that opened up insight on or gave insight on development that was not there before. And then, of course, the question arises, so if it's so similar, the molecular mechanism also similar, what makes this difference? And this is a big question that biologists still struggle with. It's not clear, for example, why the mouse is small and human is big. All these questions are still open, and you cannot find a gene for that makes the mouse small and the gene that makes us bigger, or the giraffe even bigger. Okay, because it's the same type of genes play similar roles in all these animals. So it's the organization and regulation and all this that jumps in and makes things specific, and nevertheless very robust, because human will not become a fish in the middle just because there was a fluctuation. Okay, so the guy that asked this question in a very deep way is Waddington. This is a little bit of history. He wrote this beautiful book, The Strategy of the Gene, in 1957, and he coined this concept of epigenetic landscape, asking the question about what he called canalization, meaning this robustness of specific shapes that emerge, specific morphogenesis that emerge during development. He envisioned a ball running down a landscape, much like in mechanics. But unfortunately, this is not a physical picture, because what creates this, what makes this landscape robust against all this, and what makes the ball running into this valley and not this valley is not answered. So this is a metaphor that is not just a sketch of the problem, but it doesn't help to understand the answer. And more modern approaches from dynamic system point of view say, okay, if genes are important, this is a multi-dimensional or high-dimensional space of all genes, and we should think about attractors in this high-dimensional space. And so this is a Waddington landscape in more modern form. But unfortunately, this does not help much, because, again, the number of attractors and type of dynamics is not well understood, and especially this is high-dimensional space. It's not low-dimensional space. And dynamics, I don't have time to go into that. Dynamics in high-dimensional spaces is not similar to low-dimensional ones. Now, so let me summarize the common picture that drives most of the research in this field, not only of biologists, but also of physicists, computer scientists, everybody that is interested in this problem. And the idea is that chemical patterning is in the basis of morphogenesis, and it works in what is called program-like manner, meaning there is a sequence of processes, one drive the next, in a program-like manner, and this program was set say in evolution. In practice, there are two types of concepts that stand behind this biochemical patterning. One is reaction diffusion, which I'll explain in a second, and the other is positional information. So reaction diffusion goes back to Alan Turing, the famous Alan Turing, that was, the originator of computation, and did many other things. In 52, this is his last paper before he committed suicide. He asked a very basic question. The title of the paper is The Chemical Basis of Morphogenesis. He asked a very basic question, whether there is a physics mechanism, a physical mechanism that allows to break symmetry and take a uniform egg at the beginning of embryogenesis, the development process that creates patterns. So he said it is suggested that a system of chemical substances called morphogenes. So he coined this term morphogenes for molecules that create patterns together as reacting together and diffusing through a tissue is adequate to account for the main phenomena of morphogenesis. Such a system, although it may originally be quite homogeneous, may later develop a pattern or structure. Okay, so morphogenes is still a key word in development, These are set of molecules. And the hunt of morphogenes is not very highly successful. We don't know too many of them, but some of them were identified in different species. But his idea was a profound idea because he showed mathematically that if you take two fields, U and V, that have nonlinear kinetics, and kinetics need to be nonlinear, and the chemicals of these two fields diffuse with different diffusion coefficient, and the order here is important. So one of the diffusion coefficient need to be larger than the other. Then this set of two coupled nonlinear reaction diffusion partial differential equations give rise to stable patterns. And later versions identify U as an activator that activates itself. So it's an autocatalytic process, and also activates V, which is the serves as an inhibitor. You need activator and inhibitor type of processes, and the mismatch in diffusion coefficient needs to be in the direction that the activation is local and the inhibition is global. So du must be smaller than dv. Okay, and there are many versions of this, and the success of Turing is not only in development, or not so much in development, but also in explaining all kind of patterning on animal coats and stuff like that. So there are 6 stable patterns that can emerge from Turing-like mechanism, and depending on the boundary conditions and the geometry of the system, there are different patterns that can emerge, as you see here. And these are examples of patterns in nature that are observed, that you can model somewhat successfully by applying this Turing-like mechanism, with, of course, many specific nuances depending on the system. So this is one concept that is useful. The other concept is the concept of positional information. So suppose you create a pattern of chemicals, what's the meaning of it for the biology? And then the idea was older than this, but Wolpert from 1969 developed this idea. And his famous model is called the French flag model because of this example, as you see here. And the idea was that once there are gradients created in some chemicals, in morphogenes, then cells that find themselves at different locations along this gradient, once the chemical concentration crosses some threshold, there will be a transformation of the cells to something. Once it drops below threshold, then something else will happen because it crosses another morphogene, will cross another threshold, or something like that. And then you can get patterning not only in chemical molecules, but also in differentiation of cells, which is an important step because eventually development doesn't care about molecules. It cares about development of an eye, development of a leg, or whatever. Okay, so this doesn't require any sophisticated thing. Even if you have a source and a sink, you have a gradient. But then, of course, it's a very simplistic view of what's going on, and you have to assume threshold, very sharp thresholds, in order to get a sharp patterning of cell differentiation. Okay. Now, all this is about chemical patterning, but development is not only about chemical patterning. What about physical forces? So in development, cells need to grow, move, adhere to each other, and especially a three-dimensional form should be created, and this requires mechanics. So mechanics at different levels must play an essential role in shaping morphogenesis. And this also has a long history. And my hero in this, like is an hero of many other people, is Darcy Thompson, a Scottish mathematician, zoologist. And he wrote this seminal book on Grossen form in 1917, with an extended edition he wrote in 1942. And his idea is that you can explain a lot of the forms that are made in nature, a lot of the difference between species, the way different plants, animals, whatever, make special structures by mathematics and physics. So mathematical laws and physical forces enable to explain a lot of this, and it's really a beautiful book, and there are many interesting ideas that he tried to develop. But then in more modern time, everybody realizes mechanics must be an important part of the process, but the difference between Darcy Thompson and other people approach compared to this is that mechanics is not enslaved to the biochemical patterning. It's not a follower. It's really a participant in the process. You have to integrate mechanical process with biochemical processes. There is no hierarchy that first biochemical patterning emerge and then the rest comes after that. And in that sense, the Drosophila example is a little bit unique, because biochemical pattern there certainly comes first before anything else happens, but this is not true in general. Now, morphogenesis arises by processes at different level of organization. You can break it in different ways, but certainly there are molecules like protein expression, interaction, signaling that comes in. There are cellular processes, again, that contain many different types of processes like motility and adhesion and differentiation, etc. There are processes at the level of tissues and organs. So tissues fold and organs are formed, and processes over the whole organism. But my point that I would like to emphasize is that there is no hierarchy. And the hierarchy that we assign that, okay, first molecules are doing something, then cells react, then tissue form, then this is not the way development works. It actually works by recurrent dynamics of all levels talk to all levels. Of course, not every process contains all the levels, but when you think about development in general, it's not a simple causal chain of processes. So for example, tissues can affect molecular level processes. The whole organism can, and this is taken from a perspective we wrote some years ago with Kinneret Keren that explain this. So these are for example, these are examples from mechanics, but you can think about examples from other form. There is a crosstalk across levels, both bottom up and top down. It's in both directions. That's why I call it recurrent dynamics. And there are no specific causal drivers. And this is the usual situation in biology. Usually people tell a story that this gene is doing something, or this molecule is doing something. This is many times completely misleading, because this molecule does not come by itself. This molecule, there's no molecule that can form a shape in three-dimensional space. So the entire process needs to be considered. And the challenge is that it crosses not only levels, it crosses many scales, from the molecular to the whole organism. Okay, and so we have to think about when it comes to models, we have to understand multiscale dynamics. So the usual approach, which is taken in biology, which is the reductionist approach, take a hammer, break the system into the smallest part, and start to build from the microscopics, the picture, the macroscopic picture. This is, to my mind, cannot answer the basic question we asked at the beginning. We rather would like to take the approach that is usually taken in physics, which is an effective approach, and develop some mesoscale description, which involves identify some relevant coarse grain variables, and then develop an effective description. And this is always the approach in physics, which was very successful in condensed matter physics, and it's even useful in particle physics, because always we have an effective description, and as we go down, we can use this effective description. What is special about life maybe is that there are no very clear cutoffs like we have in physics, that you can say, okay, if I want to understand elasticity of some material, I don't care about maybe the atoms or the quarks or the other energy particles. In biology, these cutoffs are not clear and certainly are not very sharp. Okay. Now, outside of levels of organization and scales, I would like to emphasize that there are three types of processes or fields. Each of them is a family of large number of sub-processes that play a role in morphogenesis. And so these are biochemical, as I said, mechanical, but also electrical. And this must all evolve simultaneously as coupled field. Why electrical? Because there is no process in physiology, not only for nerve cells or heart cells, even my skin, every process in biology involves voltage gradient and ionic currents that play important role. And this was proved in Undiling, for example. Voltage gradients play essential role in Undiling all over the place, but also in cancer and in developmental process. There are many examples where electrical processes play important role. So these three types of processes must evolve together, okay? And the coupling between them, or the integration, is one of the key questions. How does all this field integrate to achieve this robust developmental process? And the main problem is what I call here closed loop dynamics, because you can study a sub-process, but then in order to stabilize it, you have to bring in external processes, and these external processes are required in order to ensure the convergence and robustness of the process. And the point about a physical framework of morphogenesis is to try to understand the closed loop dynamics. And this is the greatest challenge. Again, this is described in more detail in this perspective in bioassay. Okay. Now, mechanical and electrical processes are really essential for closed loop dynamics. Why? Because of these two properties that they have. They both can propagate very fast and reliably over a broad range of distances. So mechanical stresses and electrical processes can really propagate fast over scales that cover all this enormous range that we discussed before. And on top of this, both mechanics and electrical processes are highly adaptive to external environmental changes and also to internal fluctuations, so they can be flexible enough to adapt and and respond to this perturbation. Okay, so both electrical and mechanical processes are organizational fields in morphogenesis. They are not byproducts or a side effects or a result of a biochemical patterning that creates the pattern and then everything else follows. Now, regeneration, which will become important in a second, I have to rush because I see that time is running. So there are two ways to get development in nature. One is fertilized egg, which is normal development of an embryo. But the other is all body regeneration. There are a few animals that actually can do all body regeneration. There are ample animals that can do partial regeneration, like, the salamandra, you cut the tail and can grow tail. In our body, bones regenerate. They don't, when bone is broken, it is fixed by regeneration, not by simple adhesion of the parts. But in this example, the flatworm planaria is a good example. You can take pieces of tissue and it will give the entire animal. So you can get planaria both from embryogenesis and from regeneration. And why regeneration is another window on morphogenesis is because the molecular mechanism and the processes involved are very similar. The molecules involved are the same. So this sharpened the question. It's not just from definite initial condition that robust patterns emerge. It's from arbitrary initial condition, also robust patterns emerge. So where is the information and how the dynamics ensure the robustness of this pattern forming system is even become sharper when you use regeneration instead of development, and this gives a lot of flexibility from the experimental point of view, because you can define different initial conditions, different boundary conditions to try to understand the physics of this process. Now, Hydra, among animals that can do all body regeneration, Hydra is the champion. The name came from the Greek legend that you cannot actually kill Hydra. It's immortal, but in fact there is an animal like that, and this is the animal. It's a multicellular animal with something like 100,000 cells, with a foot that can adhere to surfaces, a head, and tentacles. It's actually a predator. You can see it here. It preys on shrimps. That's how we feed it in the lab. So it has sensor. It has nerves. It has nerve cells. Altogether, more than 10 types of cells. It's a simple animal, but still a behaving animal, and a multicellular one with a simple geometry of a single axis of symmetry that is polar. So the head and the foot, and the rest is a hollow foot. And now Hydra played a really important role in the history of biology, because this guy, Avram Tremblay, which was a mathematician, started to play with Hydra, and he found regeneration. People didn't believe him at the beginning that Hydra can really regenerate if you cut it. But then it became clear that this is a new process in biology. And the most important contribution of this guy was these 4 memoirs that he wrote in French. This is mid 18th century, and this is considered the birth of experimental biology. Before that, biologists were just naturalists, just observing nature, and he was the first that explained how to do experiments on living systems, how to do control, how to write a lab book. So I propose my students always to read this essential book. There is an English translation to it, as you see here. that explain really very nicely and very carefully how to do experiments in biology and how to record them, and especially what is the importance of controls, etc., etc. Now, what Tremblay discovered is that if you cut Hydra into half, the two halves now complete themselves into two fully developed animals, and which have all the abilities of a mature animal. But then later, it was much later, it was discovered that even if you take a small fragment, it folds into a hollow spheroid, which develops within a short time into a full animal. But then, to the surprise of people, even if you dissociate the cells, you put it in a blender and you dissociate the cells, you take some of the cells and you aggregate them. If you have a critical mass, which is not very big, it starts to organize. Eventually, it folds into a hollow spheroid and make the entire animal. So Hydra is champion. I don't know any other animal that can do this type of regeneration. This is an example. You take a piece. This is a spheroid. This is time in hours. And you see development in front of your eyes in a relatively short time. And now you can start studying it. That's what we do for the last 7 or so years. Okay. Now, the structure of Hydra, which will become important in a second, is a bilayer of cells. The outer layer, the ectoderm, is an epithelium. The inner layer, endoderm, is also an epithelium. In between, there is a viscoelastic matrix. And then there are cables that I'll show in a second of actin that run at the bottom of the outer layer and at the outer side of the inner layer. And there are other types of cells involved, but essentially, the Hydra body is made out of a bilayer of the cells with a cavity in between that contains ions and water. And osmotic pressure, it's a freshwater animal, so osmotic pressure plays a really essential role. And because the concentration of ions for physiology must be larger than what external environment contains. And so Hydra solves this by this internal cavity of high ions that is still lower than the concentration inside cells. So this is the hierarchy of osmotic, of osmolarity. Is the lower is 5 in these units. The cells have 120 and the inside contains 60. So in principle, there should be a water flow, and the Hydra epithelium allows water flow from the external medium in and from maybe also from the inside to the cells, but mostly from the outside to the inside. And this is manifested in the spherical shell that is formed as during regeneration as the tissue folds in this sawtooth dynamics. So water comes in, the area of the tissue inflates, and then it ruptures at some point, releases the pressure, the area drops and starts again. And this is the normal thing. We can prevent this by making a small hole in the epithelium, and still regeneration will proceed fine. But if you don't do this, this is part of the natural process. In adult animal, water comes in, and there is a mouse here that the animal can regulate. So open it in a regulated way to release the pressure. Okay, so now I want to talk very briefly about actin fiber organization as part of our attempt to understand the role of mechanics in regeneration and morphogenesis. And these are two papers you can look at. One shows that active fiber orientation determines the body axis alignment, not the polarity, but the alignment. And this is done by memory carried out from the parent animal. So we call it structural inheritance. So there is inheritance through the structure, not through the molecules. And the second more recent one is on topological defects in the active fiber organization that we showed in this paper serve as organization center for morphogenesis. So actin play essential role in Hydra, both physiology and regeneration. This is the basis of the outer layer, the ectoderm. This is a transgenic animal that marks the actin cables. All the actin is inside cells, but then actin fibers connect from cell to cell to cell to cell throughout the entire animal or along the entire animal. This is the size of a cell. And to create this fibers orientation, which serves as a muscle, allows the behavior of the animal, and also stabilize the structure and plays other important roles. In the inner layer, actin fibers are organized in orthogonal direction, in circumference direction, and also serve as a muscle. So these are two organizations of muscles. One is longitudinal, and the other is in the circumference of the body. Now, the point I want to make is very short, is that when you see this or this, what jumps to mind is these are parallel arrangement of the molecular structure, and we know this organization, so it's called nematic. So this is nematic order, okay? So where objects that have somewhat rigidity align along preferred orientation. And this type of organization is described by a director field, which serves as an order parameter. And the director field is not a vector because it doesn't have polarity, but it has an alignment degree of freedom, okay? And we want to use the fact that this is a nematic, this constitutes a nematic order in order to understand the role of actin. Now, the point about nematics is that once you close nematics on a surface, it cannot be maintained, the order cannot be maintained everywhere uniformly. Defects must arise. And you know this for, if you want to cover the globe with longitudinal lines, you have to have defects in the poles, okay? Two defects. And there is a topological charge that is defined as the number of times the director rotates around a defect divided by 2 pi. So this is +1 defect, this type of defect. But you can have also others. This is -half and this is +half. And you see that the symmetry of this defect is completely different. I'll come back to this in a second. What we find out in Hydra, once the holospheroid is formed, actin, both in the ectoderm and the endoderm, makes these 3 types of defects. In the endoderm, actually, it's a vortex-like +1 defect, not a point-like, but this is in the ectoderm. So you have a point defect, point +1, -half and +half, and we can map the director field everywhere by following the alignment of the actin locally and find the defects. And we have a very good procedure now to do it automatically over the tissue. Now, the point is that in adult animal, these defects also emerge. Actually, they emerge in functional regions. So what is survived in the adult animal is +1 defect at the tip of the head in the mouse and at the tip of the foot. So these are 2 +1 defect. And then every tentacle that is going out will have a +1 defect at the tip and 2 -½ defects at the basis. And so the total charge must be 2. This is a constraint by topology, and this is always true. So if another defect emerges because of some reason, there must be other complementary defects that will preserve this charge of +2. Now the point is much deeper and interesting. I have to rush. I'll do it very quickly. If you cut a piece of a Hydra tissue, as I said, there is structural inheritance. So you see that in this flat piece of tissue, the actin fiber organization, again, I'll talk about the ectoderm, but the parallel picture emerges from the inner side. This parallel alignment of the fibers is preserved. And it actually gives memory of the original axis alignment, not polarity again, but alignment of the parent animal, the donor animal. But the point is more interesting because once it folds into a spheroid, then half of it at least becomes completely disordered. Okay, so the alignment is not just preserved, but there are large pieces that still contain enough information of the original alignment, and they once you have enough alignment in some region, it induces order for the rest of the sphere. So if this is order and this is disorder, these are two sides of the same tissue taken by light sheet microscopy. So this is order, this disorder. The idea is that this exert forces that work by dipole-dipole interaction, dipole-dipole-like interactions to induce order all around. And then when the system becomes completely aligned, defects must emerge. This is by topology. So what you see in this light sheet movie, this is development in hours, and what you see, what is highlighted are disorder regions, and then eventually you see the emergence of defects. Okay, and again, let me run this quickly. And then when the animal develops, disorder disappeared, and maybe I should look here, and you remain with defects. Not always it's easy to identify localized objects, localized defects, but this allows us to study the dynamics of defects along the way. And to make a long story short, what we discovered is that the +1/2 defects are highly motile. They can cross the tissue at relatively high rates. The +1 and the -1/2 are more or less stable. The +1 is completely stationary. And so -1/2 defects disappear by either merging with +1/2 that are motile. So +1/2, -1/2 merge and annihilate and disappear, or they do something else, but eventually they disappear from the process. But 2/2 defects, because they are motile, eventually merge into a +1. And what remains in the tissue are 2 +1. And what we found out is that there is completely asymmetry. 2 +1/2 defects that emerge early in the spheroid always become the foot, and the +1 that is completely stationary the head is organized around it, and it survives as a +1 defect. So eventually you have 2 +1 defect by 2 different processes. One emerges very early and the other is the combination of 2 +1 defects. Now they serve, the +1 defect at the end, for example, serve functional role. A mouse is created. What you see in pink here are beads that were ejected into the hollow spheroid, and they are ejected out of the spheroid by opening the mouse, which develop at the +1 defect. But before the mouse, it also emerges as opening of the tissue to release the osmotic pressure, as you see here. And it always happens at the +1 defect. Okay, and the reason +1 defect is interesting from the mechanic point of view is that the mechanical environment around the +1 defect is completely different than in the order regions of the actin. This star-like orientation of the fibers make this weaker adhesion at this point. So it calls for creation of ruptures or a mouse, and there is higher tension here that we now make some effort to understand what is the distribution of forces around this +1 defect that allows this functional, the emergence of functional features around it. So let me summarize what I showed you about actin. So the body axis alignment, there is no spontaneous symmetry breaking, as people saw before, is inherited from the parent. There is an interesting induction of order that goes by the ordered part that is remains in the tissue of the actin fiber alignment. Actin fiber nematic order is a good slowly varying field that allows exactly the mesoscopic description I was asking before, and defects in this nematic order act as organization center for morphological features. So this open up a front in research that we still in progress in our lab, together with Kinneret Keren. I'll mention this at the end. So this is for actin. So now I change gear unless there are questions about actin.
I have one question. So this Hydra, this defect dynamics of actin, how is it different from this this Dojik's microtubule?
Yeah, so
reconstituted experiment by active matter.
Yeah. So actomyosin system is an active matter, is an active system. So this is active nematics. This is not just simple nematics. And this is manifested in the way defects are formed and their dynamics. It's different from Dojik. Dojik is not a living system. It's a synthetic system. And here, the constraints coming from Hydra, I don't have time to go into it, change the parameters. For example, the fact that +1/2 is always emerges at the beginning is not completely typical to active nematics, and the motility is not the way people see motility in active nematics, and the +1 is completely stationary. So all this, there is an attempt to do theory for this, trying to extend active nematics concepts to Hydra. It's not simple because it's not clear how the two layers talk to each other and what is the role of the viscoelastic matrix in between, which can, it can store stresses and can apply forces. So it's not from the mechanic point of view, it's not a simple system. It's not a layer. It also has a structure in the third dimension that might be important. So it's not it's not a two-dimensional layer that you see only on the surface.
Well, I mean, Dojic also confined this microtubule with the kinesin on a spherical, I mean, vesicle, right?
Yes, yes, but the sphere, yes, but the sphere is homogeneous.
Okay.
But here you have two layers with perpendicular nematics and the viscoelastic matrix.
I see.
So the cry I hear from theorists is that this is not simple. The toll is on us, on experimentalists, is to understand what is really important in all this mess. I mean, and but there are features that deviate from what Dojic observes, and it's not clear what is the reason for these deviations. But again, the point is not, I don't think that Hydra is a good material system to study active nematics. I'm saying the opposite. I'm saying that if we want to understand morphogenesis in the context of Hydra regeneration, and this is very general because actin plays a role in almost all, actomyosin plays a role in almost all developmental systems, then for Hydra, going into the nematic description gives us an ability for a coarse grain field that we can start to study as an organizer of morphogenesis. Okay, so I don't like, Hydra is an active system, but it's not the best simple active system to study active nematic, but it's indeed active nematics. Okay, maybe I should go on.
Okay, probably I'll ask the question later then. Okay, because
you can, I mean,
right now, okay, I think it's relevant a little bit.
So what about? Sorry, we can either do question now, and then the talk will be longer than, or you'll stop me, whatever you want. Yeah.
Okay,
please
I have a very short question. So if you put the Hydra in the kind of extreme solvent condition, so right now probably like a polar solvent, right? But if you put it extreme apolar solvent, what happens? So that was a simple question, but if you think it's not proper time, then we can do it later.
You mean to get rid of water?
Yeah, because Hydra is so much probably active.
No, it's an interesting question, but I don't know if life can sustain. Be sustained without water, so we never tried it. So
yeah, thanks. We can continue.
Now, so now we change gears. I'm going into the central subject of this talk. Under the question, is it possible to modulate morphogenesis on demand? And the importance of this question, so it could sound like a funny question, stupid question even, but it's not completely stupid because I think we can do it. But it's a little bit more deeper than you think, because if you can control morphogenesis on demand, then it's a strong deviation from this program. And especially if we perturb the system and control the stages in morphogenesis, then different parts need to reorganize, and it highlights again the question of the robustness of the integration of all these processes to give you at the end a viable animal. So we try to do this by two means. The idea is to utilize external physical fields, either mechanics, and there are many tricks you can play. For example, you can make Hydra magnetic by coating it with, that's the beauty of Hydra. You can really torture it and it still regenerate. For example, you can coat it with magnetic beads, make it magnetic, put it in a magnetic trap and start pulling on it and try to define it. We grow it in channels. And we try to see if we can, like in a clay, create a body form that will direct morphogenesis. But more interesting for this talk is that we found out that external electric fields are highly useful for as a way to control morphogenesis. So the rest of the talk will be Hydra regeneration under external electric fields. And this has also a long history. This guy Rouse in the 19th century, beginning of the 20th century, tries to put electric fields on eggs, develop a fertilized egg to see if it can affect development. It was not successful. Later in the 20th century, Robert Becker and others, he wrote this very nice book. They suggested that electrical processes, both in physiology and regeneration and development, must be important. He was an orthopedic surgeon, and he was bothered by the fact that people didn't understand that bones really regenerate, and started to study these processes and other processes to show that, that electrical processes are important. And Michael Levine in Tufts in the US is pushing this direction, mostly on planaria, but not only, to show that if you perturb electrical processes, you can get aberrations or modifications in morphogenesis. So even a brief blocking of gap junctions, we'll come back to it later, can create animal with two heads, for example. And this can be also memorized to later generations. And the idea is that electrical processes are important. But, I should mention this is an Israeli company, Novocure. This is the best cancer treatment you can find today that treats brain tumors that are completely fatal. So these people have 2 months to live or something, and there are already people 5 years after the emergence of cancer that it's completely disappeared and didn't come back. And the treatment is simply putting alternating electric fields on the head at these frequencies, hundreds of kilohertz. And this is so successful, this treatment. It shows that physical treatment might beat genetics and all this. Maybe we can find something for corona, I don't know, that is completely physical. But in that case, the FDA stopped the experiments in the middle because the effect was so immediate and on so many people that we saw that the control group is not a moral thing, not ethical. The treatment should be applied to everybody. And this is now in completely clinics. It was approved. And so now Hydra is special from the electrical point of view in the sense that all the cells in Hydra are electrically excitable, meaning that if you perturb it electrically and cross a threshold, it develops an action potential, much like neurons. So it's not only neurons in Hydra that are excitable, there are neurons, but also the epithelium is completely excitable. Remember, it works like a muscle. So it's not highly surprised that all these cells also respond to electrical processes, but this gives us an opportunity. So this was shown long ago, that Hydra can fire action potential from all over the place. And even if you make, there is a protocol to make nerve-free Hydra without nerves. And also you can get electrical excitations in the epithelium. Now, this for us is great because then application of even weak electric fields from the outside get amplified by this internal nonlinear excitation, excitatory or excitable dynamics. And so there was a hope that we can, by external electric field, get excited results. This is the experimental setup. We make, there is a substrate, we make holes in gels that allow Hydra tissues to float free in 3D, but will not escape from this location. and each well is located between two electrodes. The system is under wash, continuous wash, to make sure that the medium is in constant condition. And the way we can run the system now, for each sample, we can give a different voltage and frequency by using a multiplexer that runs on the system, and then time-lapse microscopy allows to take continuous movies in parallel to the electrical, the application of electrical fields. So this is an example. I'll run it fast because we're running out of time. So this is a zero voltage. The spheroid develops, you see here, tentacles and everything. Then we increase the voltage, here above 20 volts. And in the system, go back a little bit, and the system becomes spheroid again. You release the voltage. I'll show you it again in a nicer way in a second. And it develops again. You see it here. You increase the voltage again, it becomes a spheroid. So you can control at least the form of the animal, and there are two routes in the experiment to go. You can either, so there is a critical voltage. Certainly if you go at low voltage, it doesn't do much to the system. There is a critical voltage. If you hold the system above criticality from the beginning, it will not develop. I'll show you in a moment. But then if you release the voltage, it develops. So the point is that the regeneration potential is not lost in the process. You just hold it. You can go through another route. You can let the system develop, go above criticality, and then it folds back, and then you release it to make sure that you don't do any damage to the tissue. You just deform it, deform the process. So this is an example of going in the upper trajectory. You put high voltage, it attempts to start to develop, but then it cannot overcome the external perturbation. Then you release it, starts to develop. And here you go, it develops, you go above criticality, and then release it to see that the potential, the regeneration potential is preserved. Let me, this is, for example, starting above criticality here. You see that the system attempts to start to develop, but it actually cannot. It does not develop even for a long time. You release the voltage and for the same experiment, and it starts to develop. Then you apply higher voltage, and again, it does not, it falls back, and then you apply the zero voltage again, you remove the voltage, and it starts to develop. Okay? There are many interesting things you can ask about it. One thing that becomes clear is that if you do rounds of going backward and forward around this critical point, you have to increase, there is adaptation of the tissue, and every round you have to increase, the critical voltage is increased. This, by the way, is statistics of the many samples. It's not a rare event. The problem is that if you go with voltage too high, it can disintegrate the tissue, and this is irreversible. But if you do it carefully enough, you can get for every sample, if you do it carefully enough, that it first develops and then folds back. This is just a cumulative distribution of many samples to show that given enough time and high enough voltage, you can fold any of this, you can refold or reverse the morphogenesis process. And the interesting stuff is that in many of these samples, actually in most of the samples, if you hold the system you halt regeneration, then once it redeveloped again, start regeneration again, a new body form is formed. So it's like initiating the developmental trajectory, a different developmental trajectory. You see here 2 tentacles, here you see 4, here you see a very deformed body plane that after folding it and reinitiating the regeneration, it's fixed into a normal appearance of Hydra. Okay, and you can do rounds. The record is 5 rounds of going back and forth. At some point, the system loses energy. There is no energy supplied, just basically in water. So it loses resources, and at some point it becomes harder to do this. You can do 5 rounds of going back and forth around the critical point. Now, calcium plays an important role in all physiology, certainly also in regeneration. So we made a strain with a calcium probe that you see here. This is a fluorescence image of the spheroid. This is at 0 voltage. I'll come back to high voltage in a second. This allows us to study the calcium dynamics in this spheroid. And if we can halt the system for a long time, we can gain a lot of statistics. Let me go fast. If you go to high voltage, it starts to be highly excited and it falls back. Now, why calcium? So calcium is an important ion. Calcium 2+ is an important ion that plays an important role in physiology all over the place. But for us, it's also important because it's a universal effector that also mediates between electrical, mechanical, and biosignaling. This is the junction we are interested in, to look on the junction of electrical, mechanical, and biochemical processes. So calcium is really a good indicator. And again, let me rush quickly over this. At 0 voltage, these are spikes of calcium activity. There are 2, I put 2 experiments, one on top of the other, just that you'll get impression of the differences. You increase voltage, you see that the density of spikes get higher. And then if you go above criticality, which is 20 volts here, you see very high activity of calcium. Okay? And you can gain enough statistics to see that indeed above certain voltage, the calcium activity that you see here, the fluorescence, goes up by much. This is logarithmic scale. So the extended tail of activity that you see here in the distribution shows that the activity in general goes up very much under voltage, under the external voltage. I'm going to skip a lot of this. I just want to mention that calcium activity is directly connected to electrical activity. We showed this by 3 types of processes: direct electrical measurement that I'll show you in a second. We showed that there is a strong upper frequency cutoff. So if you go above 3 kilohertz, remember the cancer treatment was at 100 kilohertz, so it's a completely different mechanism. Here, if you go above 3 kilohertz in the AC field, it becomes transparent. You can put whatever voltage you want. The system is transparent to it, but below it, it works. And this kilohertz times frequency corresponds to millisecond timescales. So it corresponds to the RC timescale of the tissue. And also you can create high excitation of calcium by enhancing the external potassium concentration, which is a standard way to do it. Just let me show you the electrical excitation. So this is a parallel measurement of fluorescence of the calcium and direct electrophysiological measurement by an electrode. This is a burst of action potentials measured directly that you see them here. And every burst of calcium follows actually a burst of action potentials that are electrical excitations of the tissue. So this is just to show the frequency cutoff, but I'm going to skip this because of lack of time. Now, the question is whether this is the reversal morphology is the only thing that happened, and the short answer is no. So the head organizer is an important, I'm going to skip the history, but the head organizer in all developmental processes is a very important concept, is a group of cells that induce order in development. In Hydra, the head organizer is a group of cells of something like 50 cells that sit at the head, at the tip of the head, and induce direction and polarity of the axis, and allows Hydra to renew all its tissues all the time without losing the body plane. So it plays a really essential role, again, in all developmental systems, but especially in Hydra. It was found out that from the molecular perspective, it's the same mechanism, again, across species, the one WNT system. It's an autocatalytic system that maintains itself. You see here dye at the head, at the organizer at the tip of the head. This is a bud. The emergence of a new Hydra also shows this thing. And what it showed that if we put a voltage, again, let me, so this is a transgenic animal that the organizer is marked by GFP. And if you increase voltage, the GFP disappears. Like you see here. On the right, you see a marker in red that shows that the fluorescence does not go away. It's not because of imaging or anything like that. And then when you release the voltage, you clearly see here that the organizer comes back. Let me summarize this. This is an average over 7 samples showing the decay of the organizer upon increasing the voltage above criticality. This is a specific trajectory for one of the tissues. You see the organizer very clearly. And then even if you look on all directions for a long time, you don't see fluorescence. And then you release the voltage, it starts to come back, as you see here. Now, why is reversal development an exciting surprise? So the word development actually mimics the way we think about developing films. That's the way people thought about it, is that developing a picture captured by a camera film. So it expresses the fact that development is an unfolding of some information. This is a very long and interesting history that went under the name of preformation, replaced later by epigenesis. And any idea, all these old ideas will turn out to be completely wrong, of course, because there is no small creature in the embryo that is developed by the process, unfold by the process of development. Rather, there is a dynamical process. But for me, the thinking of a program is much like that. It's all the information is contained initially in the embryo. Now let's run the program and it will give us the process. Now reversal development, of course, if parts of development or all of development is reversible, then it destroys this picture, because either you break parts apart, you reverse part of them, but then when you release it, they need to accommodate to the rest of the system that was running ahead, or you stop most of the essential processes, and then you can reinitiate them from arbitrary initial condition. And the question, how you preserve all these things in memory, is not the right question. It's the same surprise that people had thinking about stem cells and the emergence of differentiated functional cells, and then the discovering of what is called reprogramming, that you can by perturbation take a functional cell, say a skin cell, and drive it back to become a stem cell with the potential to become anything, any other cell type. So this on the level of a cell is now completely accepted, although in vivo it was not sure, it's not clear that it was proved, but in vitro people know how to do it. The question whether you can do it for the whole animal in development, you can run backward the process, sounds like a crazy question, but it's a completely open question for me. Especially you can run part of the processes backward, and then again it's a deep question about development, how the rest reorganize to accommodate this strong perturbation. So can I have another 10-15 minutes, something like that? What's the situation?
Yes, go ahead, please.
Okay. So now what I want to do, so this is, now I want to use the fact that we can halt regeneration at a point, especially at the edge of regeneration. So if I release the voltage, regeneration will start from this point to study fluctuation and correlation near this critical point at the edge of regeneration. Okay. So again, the motivation for this is that I want to get a framework that deviates from this program type of idea, and the fact that people think about that what stabilizes developmental processes, morphogenesis, is by threshold crossing of cellular processes like the one I showed when discussing positional information, stuff like that, and by development of symmetry breaking events and fields, halting regeneration allows us to deviate from this. And so it provides a new methodology to start to understand this fascinating process of development. In that case, in the context of Hydra regeneration. Now, this is highly non-trivial to study. So I want to study the calcium dynamics as a first step towards this direction by halting the process and look on fluctuations and correlation in the tissue. Now, this is highly challenging because this is a tissue that is free in three-dimensional space. I don't want to localize it because this will imply a mechanical constraint that will affect the dynamics. So it needs to be free. And this is not this is not the way neural system or cardiac system in culture or in vivo studied, because usually people fix the tissue in labs in the lab space, in the lab system, and so there are coordinates for each cell, etc. Here, the tissue is free to rotate. So how you analyze free to rotate, free to move, free to change shape, and also it's a deformable object. As you see, it can inflate, it can develop aspect ratio that is dynamic, etc. How you study this. And so the idea is to use statistics instead of trying to follow each point, the dynamic at each point a long time, use the time as an ensemble and look whether we can find universals in the statistics that allows us to to understand the dynamics in the system. To this end, at each time point, I build a polygon around the tissue. I shrink it a little bit to get rid of edges, and then I build a mesh on the tissue using this polygon that allows coarse graining of the fluorescent intensity in pixels according to in a controlled way. So now each triangle here in the mesh gives me an average signal of the pixels inside it, and now I can ask questions about correlations and statistics and fluctuations in this mesh. And of course, there are statistics that do not require a mesh, and I compare with mesh and without mesh to see that the mesh does not give any deformations in the information we get from the signal. Especially there are no overlaps, there are no pixels that appear in two of these coarse grain triangles. Now, the experiment goes by running a voltage protocol of the external field. You can do all kinds of protocols and study the voltage dynamics. For example, this shows the two first moments in the calcium intensity, the mean and standard deviation, the mean in blue and standard deviation in red. I'll come back to a better experiment in a second. This is just to show that mean and standard deviation, this is the spatial standard deviation. For each time point, it's a fluctuation in the tissue, the level of fluctuation. And this is a logarithmic scale. You see here that the standard deviation, although it follows the mean, it can give you different dynamics. Actually, it gives you more information about the dynamics. Okay? And what we found out, I'll expand a little bit on this in a second, is that actually you want a measure of the fluctuations, and the right measure is the standard deviation over mean. Not the mean itself and not the standard deviation by itself, by the standard deviation over mean. And what you see in this trace, this is a time scale in minutes. This is an example. In black, you see the area of the tissue. In blue, you see the aspect ratio. So these are the two moments of the morphology, if you want. It's the area and aspect ratio. And in red, you see again the fluorescence intensity, standard deviation over mean, the fluctuations. And the left side is at 0 voltage. The right side is at high voltage that halts regeneration. And you see immediately by eye, you don't need sophisticated sophisticated measures to see that something dramatic is changing when you apply the voltage. Both the area and aspect ratio start to fluctuate like crazy, as well as the level of fluctuation, the standard deviation over mean. There is a high frequency in time that starts to play a role here. But there are interesting correlations that I'll come to in a moment. And at 0 voltage, it's much more relaxed, but still there are peaks in standard deviation over mean that are interesting for the morphology. Okay, again, you can take 0 back, the same tissue back to 0, and then you can go to higher voltage. Here is 50 volts, here it was 40 volts. And you see again, very high frequency fluctuations of the aspect ratio and the area, and the fluctuations of the fluorescence becomes also highly fluctuating in time. There are two types of fluctuations here. Okay, there is, for any time points, there are intensity fluctuations of the tissue. So it's the level of variance, if you want, normalized by the mean, of the signal at each time point. And a long time, there are temporal fluctuations of this signal. Okay, so there are three measures. Again, standard deviation of the mean, the spatial variation of the tissue at every given time point, the tissue area, and the aspect ratio. You can compare this and this. You see that voltage increase changed the dynamics. And again, because of adaptation, I have to go to higher voltages to keep holding to take the system in reverse and holding it at the edge of regeneration. You can take a different approach. So our understanding coming from this was that the way parts of the tissue talk to each other is important at every given time point. And the main electrical communication in Hydra is through gap junctions. So it's proteins that connect each cell to neighboring cells and allows ionic currents to go through it. So the question arises, what happens if you block this gap junction? We use a small alcohol molecule that blocks gap junctions in Hydra very well. So when you block gap junctions, you can halt regeneration. And you can halt regeneration in a reversible way. Again, you wash the blocker, it starts to regenerate again. At this point, it regenerates. But now you can study this critical point of halting regeneration from a different perspective, not from holding the external voltage high, but blocking an internal process and compare them. So you see here that the system will not develop for a very long time. You can hold it as long as you want, like with the voltage. Once you release it, it starts to build order. And what is dramatic is that a change in the calcium behavior, you start to see spikes in the fluctuation, the standard deviation over mean of the calcium dynamics that is completely different from the previous session where the gap junctions were blocked. Now you can take these two approaches, either the external voltage or the block gap junction, and start to ask questions about correlations. And so if you look on the cross correlation, so the first thing I want to show is that the fluctuations in the calcium dynamics actually is highly correlated with morphological features, with the mechanics. So it's not, the standard deviation over mean is indeed a relevant measure or relevant variable to try to understand the connection between calcium dynamics and morphogenesis, the emergence of form. You see here an example. This is at 0 voltage, this is at 40 volts. You see that it can go in either direction, either positive correlation or negative correlation, but it's highly correlated with morphology. In that case, is the area. The gray in the background is a surrogate. We take the time trace and randomly mix it to make sure that the correlation disappears. And that's what you see in gray here. Okay, and what you see on the right, this is a scatter plot that shows you exactly what you see here. The red is positively correlated. So this is the area, and this is standard deviation over mean, and the blue is negatively correlated in that case. You can find other examples where it again, for example, in the blue here, the correlation at 0 goes to 0 at 0 voltage, but it develops a time lag. So the dynamics can be much richer than just direct thing. And especially I want to point that there is a time scale of 100 minutes that appear in the system that we see all over the place. I'll show you in a second. The aspect ratio is also correlated with the dynamics. Correlation is weaker in that case, but it still exists. You see it here. And you see even oscillation. I'll show you in real time this oscillation later or momentarily because we don't have enough time maybe. But you see that these are real correlations. Again, this is the surrogate that shows oscillation with a time scale of 100 minutes that appear in the system. So both aspect ratio and area, where the area is a little bit stronger correlations, are correlated with standard deviation of the mean, the spatial fluctuations of the calcium intensity. Actually, to get an impression, you can see this synchronized movie. This is bright field on the left, and this is the fluorescence on the right. Look at it for a minute. You'll see by eye that fluctuations in calcium that comes in affect the morphology of the tissue. This is an example at some voltage. And you see patterning appearing in the calcium dynamics, which is manifested in the fluctuations, in the standard deviation over mean as a measure of these fluctuations. Okay, so this is the picture from the calcium dynamics. You can ask more generally, what is the distribution? This shows the variability among tissue. Each tissue is an individual in biology, and it has its own signature. And also the voltages affect the statistics. So this is a distribution of the standard deviation over mean intensity. This is under this voltage protocol. You see different distributions. This is a half-log scale. And you see immediately that it's a skewed distribution. It's not Gaussian. And what is really interesting, but I don't have much time to go into it, is that if you take all this distribution and you normalize them, let me explain the normalization because it's a little bit tricky. Standard deviation over mean, it's a number at a given time point. It's just a number. Okay. Now think about the time trajectory, the time trace as an ensemble, statistical ensemble. This statistical ensemble has average, which is marked as mu here, and standard deviation. This is in the ensemble, it's sigma. You take the standard deviation over mean, call it x here, normalize it by subtracting the ensemble average and dividing by the ensemble standard deviation, all this distribution collapse to a single curve. It shows that the shape of the distribution, although the distribution in physical units are different, in normalized units, all the shapes collapse to the same shape. And interestingly enough, the best fit to this shape, which is deviates, it's not Gaussian as you see here, it's skewed distribution with an exponential extended tail. The best fit is part of what is called generalized extreme value distribution. Specifically, this is the Fréchet distribution. Okay, again, so important point is that the standard deviation of the mean, the fluctuation, is the only dynamic variable that collapses. This is the mean, it does not collapse well. This is the mean, it does not collapse well. This is the variance by itself, it does not collapse well. The standard deviation of the mean collapses well. Even if you take the gap junction blocker distribution, which is completely different, looks like completely different shape in real space. If you normalize it, it collapses to the same distribution, which is given by this function, is a well-known type of distribution. So there is a lot of interest trying to understand why extreme value statistics has to do with this process, but I'm not going to discuss it now. The variance and the mean are highly correlated, and the variance versus mean for all the experiments that involve voltage go as a mean to the power 2.5. So in Poisson process, I just remind you, variance goes as the mean. Here it goes as the mean to the power 2.5. And when you block gap junction, it goes to the power 3, more or less. Nevertheless, they all show the same distribution, but variance and mean are highly correlated. It means that the standard deviation over mean itself is a function of the mean. It's not a simple function of the mean. It goes up with the mean. Okay, let me, another 5 minutes, it's okay? I just want to show something about more about the correlations. If you look on the autocorrelations, again, you can see oscillations. You can see correlation over non-trivial timescales. This may be a cleaner way to look at it. You see clearly that under certain conditions, the tissue standard deviation over mean, the fluctuations start to oscillate in a regular way. And the time scale involved is around 100 minutes. And I think I'm going to skip this to leave time for questions, but because this is a little bit complicated. But if you do careful analysis, you can show that the calcium activity is localized. Now, if you don't block gap junctions, localization of the calcium activity is highly surprising. If you take, for example, cardiac tissue, the heart, which is similarly excitable tissue like the Hydra muscle, if you excite the heart at some point, the excitation will propagate everywhere, unless there is a defect. This defect, of course, is it can be fatal in physiology, can lead to fibrillation and all kinds of other disasters. But if the heart doesn't have defects in structure, even if you take heart cells in culture and let them connect to each other, any excitation that you do in the system propagates across the entire system. And this is due to the gap junction connection, which is a very strong connection of the electrical activity across the simple. Very surprisingly, in Hydra, the signal is localized. So you could find repeatedly just an island of activity that keep flashing. The rest of the system can be silent, and then another island. And then you ask about the correlations, spatial correlations in the tissue. Again, this is a spatial-temporal dynamics. It's very hard to measure it. But what you see is we divide the tissue into bins and ask what is the correlation, if you want the covariance, normal covariance is the correlation at different distances. So you take each pair of points that are, for example, 0.1 to 0.08 to 0.12 in distance, all the pairs, and you compute from this the covariance matrix, normalize it, get the correlation at this distance. Now, because the tissue is deformable and can change area and shape, you need, in order to compare different time points and different samples, you have to take these bins as a fraction of the maximal distance. When you do that, these are distributions of very large statistics over many samples that are taken at zero voltage, high voltage, blocking gap junction, etc. This is at this bin of distances, this is at this bin, etc. If you follow, for example, the peak of the red, which is at high voltage, you can recover correlation function, an average correlation function for this voltage, if you want. But for me, it's better to look on the entire statistics at each bin, because you see that it's not, there is a distribution, there is a, and indeed you see differences between high voltage, zero voltage, and block gap junction, okay, which I summarize here. So what's the difference between halting regeneration and holding it at high voltage, and halting regeneration but blocking gap junctions? There are two differences. One is the fluctuations of calcium are much larger with voltage than with blocking gap junction. And the second, you have much higher correlations, spatial correlations in the tissue at high voltage. So when you excite the system by applying voltage to the tissue, different parts of the system talk to each other in a synchronized way. When you block gap junction and a fluctuation arises in the tissue, it hardly can excite fluctuations far away from it. And this leads to a picture of the reason these two different mechanisms halt regeneration or halt the morphogenesis is, and is summarized here that you, let me summarize it here. There are two opposing forces in the system. One is the osmotic pressure that gives tension to the tissue and wants to inflate it. And there are the internal forces by the actomyosin system. This is the active part of the system that opposes the osmotic pressure and can be directional. Okay? Now think about rowers in a boat. If you have many rowers, you know, in Olympic Games, what is important is the coordination between the rowers, not the force that each one of them is applied. And the same is here. If you have high correlation between parts of the system, then you can build a coherent internal force that will start to pattern the morphology. And in high voltage, you have this coherency, but what you have is also high fluctuations. So if you average over time, you see that for the time scale of 100 minutes that the system needs to rebuild anything in the mechanics in it, there is not a coherent force over this time. So although there is spatial coherency in high voltage, there is no temporal coherency that allows this. In gap junction blocker, there is no spatial coherency. So in both these cases, there is no way to build a coherency of the active forces to pattern the system. Now, let me summarize very quickly because I went over time by much. First, standard deviation over mean is important. Fluctuations are important. That I said. And I want to summarize the entire talk by a few sentences. So first, there is a critical electric field below 3 kilohertz that halts regeneration and even leads to reversible morphogenesis. And it's important to emphasize that the revert tissue keeps its regeneration capability. So there is no damage to the tissue. Each time you restart regeneration, you can initiate a different trajectory in development. This is manifested in a different body plan that emerges every time you do a cycle like this. The process of reversal morphogenesis is triggered by enhanced electrical excitation, followed by enhanced calcium excitation that in turn affects the mechanics the way I showed. And this electrical mechanical coupling is the process that leads to reversal, but nevertheless, it's not manifested only in mechanics. I showed you that the central signaling system also decays under this condition. And the implications are very wide. We need to extend the picture of development beyond biochemical, certainly beyond biochemical patterning, forward-driven hierarchical processes. Clearly, electrical processes play an instructive role, so we have to integrate them into the process. So they are tightly integrated and coordinated with the rest of the types of fields that I talked about, the mechanical and biochemical. The question whether the developmental process is fully reversible, namely differentiated cells go back to stem cells, it's far in the future to show how deep is this reversal process, but nevertheless, there is a lot of interesting questions that we can start to answer. Everything about the mechanics and the polarity that is done in our lab is done actually in strong collaboration with Kinneret Keren, and the work on defect was led by Yonit Mardus Sachs, a student of us, and mainly in Kinneret's lab. And these are the rest of the students working, or some of them left already, worked on this project. And Gidi Ben Yosef is our technician. And the electrical measurement was done, the direct electric measurement was done by Hillel Ori, now a postdoc in Harvard. And we got transgenic Hydra strains from these guys. The calcium strain was a strain that was constructed by us, but the rest of the strain used for the Actin and for the organizer are by these labs. So thank you very much, and I'm sorry for the time.
Thank you. Audience, it's time for discussion. You can make a voice if you have a question.
Can I stop sharing or what?
Now,
I think it's okay to keep it like that.
Okay.
Professor Che, is there any questions or not? Then I have a very simple question to start.
Go ahead, please.
Okay, thanks very much for your talk today. I have a just simple question about whether you can actually map between electric field strength versus temperature. You see what I mean? 10 kilovolt or 10 volt into the cell.
I think the temperature, you ask about temperature, right?
You know, yeah, temperature, how much do you expect as compared to 40 volt?
No, so temperature fluctuations are reduced here by, we flow medium, fresh medium all the time that is kept at constant temperature. So as I showed in the setup, the medium is replaced continuously. And I think there are temperature fluctuations that are important here.
Okay, so controlled by room temperature, everything.
Yeah.
Okay, I see. But the second thing is the electrode polarization. Do you expect some kind of building up charges onto the actually 30 kilohertz or something like 3 kilohertz level?
No, because you know, people try. We can get some effects by DC, applying DC voltage, which is not a good idea, because DC voltage change the pH and other things very quickly and accumulate junk on the electrodes. But people try to find charges, morphogenes that are charged in many animals, also in Hydra. And in some cases, in Hydra, there are no charged processes that you can affect by simply polarize external field.
I see. The final thing is so calcium activity. It's wonderful to see actually the consistent measurement. And I wonder that activity may be related, you can divide it either stretching or shrinking, like a contraction. What do you expect that the most activity of this calcium-driven head organization of Hydra? Do you think that you see more trade of contraction versus stretching?
No, but it goes in both directions.
Both directions.
Look, for example, here, this is just zoom over one region. You see area fall, so it shrinks. You see increase in calcium activity. You see the area goes up. You see also increase in calcium activity. It can go both direction. That's why the correlation, cross correlation, can go negative or positive depending on the direction. So it's not just one way. Any change in morphology, actually either area or aspect ratio, accompanied calcium activity. And in many of the cases, calcium activity precedes the morphological changes, but not always. And in many interesting cases, the calcium activity dynamics itself is get feedback from the mechanics. So you shouldn't think about it as a sequential type of processes. It goes together. Okay,
then that leads me the final question. What if you play magnesium? Because as far as I know in the biology, that calcium and magnesium, they somehow counter interfering. So when you play here in this system, magnesium maybe have kind of an alternating component to play.
Yeah, magnesium is very low. Of course, magnesium is essential in physiology, but it's very low here. I don't think it plays. Calcium itself is a very important ion. So
okay, I see. Thank you very much.
Okay, and let me read one question from audience in the message. What is the best method to identify the topological defects?
No, so the method we used here, I think there is a reference, but if not, is taken from identifying fingerprints. In fact, I don't think I have it here. No, so there are methods that are developed for pneumatic system, but the one we use is an algorithm to identify actually fingerprints. So fingerprint is always a pneumatic, as pneumatic order in it. And so there are good algorithms. If you go to this paper, not this one, second. If you go to this paper that was recently published in Nature Physics, this method is described in detail. But it's not ours. I mean, it was taken, it's just a modification of known algorithms to find order in parallel array systems. So there are good algorithms to do it. But this paper describes all the details.
Thank you. Is there more questions? If not, I have one question. Can you give just comments, general comments, the role of microtubule on the morphogenesis?
Okay, so microtubules also play a role, but they are not. So microtubules in Hydra will not study, very carefully. But there are indications that they play a role in regeneration, but much less dynamic than the actomyosin. You see, basically, because Hydra is a muscle, okay, is really basically a muscle. That's what I showed you, right? It's basically a muscle, And it's muscle not only after finishing development of an adult organism, even if you start from a tissue and look on the tissue, how it reorganizes, even if you look for aggregate of cells, the first thing that develop are two layers of epithelium that are not symmetric and actin organization inside them. So long range actomyosin organization like this is by the way not the only actin in cells, because the cells have also, you can see it here, but this is at a different layer. This is the normal actomyosin that you see in every cell that is part of the cytoskeleton that supports the skeleton of the cell itself, the shape of the cell. And these fibers reside in a lower layer, again inside the cell, but at the lower side of the cell, and they're completely different. And microtubules reside only at that layer. And so they play a role of shaping forming the shape of cells, delivering forces and stuff like that. That certainly they play a role. But for morphogenesis, the actomyosin is much more dynamic, and I think it's much more central to the process than microtubules.
Thank you. So there's one question from the Professor Kim at Jeonnam University. Can you make a voice?
Okay. Can you hear me?
A very small sound.
Okay.
Can hear you.
Okay, I raised my voice. How about now?
Very okay.
Better?
Yeah.
Okay.
Still small. Can you be a little more loud, please?
Okay. I don't know what happened here with my microphone. Okay, so how about now?
Yeah.
Okay. While I was listening to your nice talk, I was thinking probably it would be critically useful to come up with some Langevin type stochastic model to describe voltage dynamics of yours. However, after hearing your presentation about fluctuations and correlations, I realized that it will be very difficult to write down, for instance, autocorrelations and so on. So I gave up my thinking of this direction. However, my question is, what about thermodynamics? In other words, can you consider Hydra as a system, non-equilibrium, and think of applying or exploring fluctuation theorems? It seems that your voltage dynamics has a well-defined protocol. So have you thought of this? Or you already proceeded with some thoughts?
Sorry, I barely heard.
Oh, sorry.
Maybe
he's
you asked about fluctuation dissipation?
Yeah, non-equilibrium. No, not fluctuation dissipation. Non-equilibrium fluctuation theorems.
Fluctuation theorems.
Right.
Well, I don't know. I mean, fluctuation theorem is a general name for active systems that are out of equilibrium, I guess. I think that if you want to me, integrating electrical, mechanical, and biosignaling needs more than just a simple, say, Langevin type or something approach, unless you want to understand something very specific in the system. But for example, to understand even to understand the fluctuation in calcium, forget everything else. Forget regeneration forget morphogenesis. Even to understand the fluctuation of calcium require more because it's a spatial temporal system. You have both space and time.
Yes.
And so to understand why spatial fluctuations in the tissue behave a long time the way they behave, why they give statistics that belongs to the extreme value universality class and stuff like that, you need to come with an idea. I don't know if fluctuation theorems help or not on this. I'm not an expert on fluctuation theorems, but I guess maybe it's better to think what is the question first and then try to answer it. What you want to explain?
Okay. Can you hear better now?
Yes,
Yes.
better.
Okay. So forget about Langevin equation. So you apply electric field to your Hydra as a system, and you have well-defined protocols for how to apply electric field or voltage. And I saw your data. So your Hydra was at the initial state, and then it changes. It changes not only morphology, but also its thermodynamic state. That is my question. So if you are interested in, no, I'm sort of suggesting, if you're interested in how the non-equilibrium thermodynamic states are changing as you apply Visual electric field, the current non-equilibrium fluctuation theorem may be a good tool to investigate such an idea.
Yes, but you see maybe I should say something about, I don't know about fluctuation theorems, if they are good or not, but let me just mention one thing. Hydra is an excitable medium in the electrical sense, which means that if you look just on the epithelium layer, it consists of cells that are highly nonlinear in response to electrical stimulation. This is one thing. The other thing is that the system, in principle, if you don't perturb it, it's strongly connected through this gap junction. So think about, I don't know, maybe the heart tissue is a good example. Although the Hydra is more similar to smooth cells than cardiac cells. But if you think about, if you take a piece of cardiac tissue, you understand what I'm saying? You take a piece of heart cells and you excite it at a point, either mechanically or electrically, this propagates all over the place. It's not localized. Now, Hydra is a special, I think I wrote it here, maybe I skipped it. What is curious about Hydra is an active system from this perspective of not the actin, but let me just 1 second. See if I leave it here. So it's written here, you can read it. Unlike other electrical excitable systems, every excitable system that I know of will not behave like that because the activity will not remain localized. Again, think about the heart. If the activity becomes localized in the heart, this is a huge failure of the tissue because it needs to be coordinated. Now, in all muscles that I know of, and Hydra is a muscle, there is a strong coupling in the system. So these two features, the fact that the system is is excitable electrically and strongly connected suggests, as I wrote here, a different type of active matter. Okay? And the interplay between local activation and spatial patterning is highly now, if you can expand some of this by fluctuation theorem, it's okay, but take into account that the fluctuations are non-Gaussian, as I showed, and this extreme value statistics keeps bothering me because it shows that there is some mathematical relation between the fluctuations, if you want, some constraints that give the same shape of the distribution under very different conditions. And this is very general. It means, you know, it's the analog, it's a parallel to get a Gaussian distribution for many processes that belong to the same class of, say, Brownian motion. So on one end, once you see a Gaussian, you cannot reverse engineer it and get the process because it's universal. On the other end, it tells you that it belongs to this class of processes. Every time you see deviation from Gaussianity, you should ask yourself what is the mechanism that leads to this deviation. And here, the deviation is not arbitrary. It leads the distribution to collapse into a universal class that is well studied, but not well understood. And this is the class of extreme value statistics, which means that if you have a series of processes, you take the maximum or minimal, the extreme, and you look on the distribution of the extreme out of many repeating processes, you get a distribution like the one I showed, which is always skewed, it's non-Gaussian, and in some classes it has an exponential tails, in some classes it has a power law tail. In our case, it's an exponential tail, at least for the range that we can measure within the system. Is that touching your question, or maybe I'm...
Well, I understand. Thank you very much. Simply, I like to just conclude, close my question by saying that probably this another aspect that I'm suggesting. If you
Okay.
are interested in exploring thermodynamics in your system in the future.
Yeah, it might be very useful, but I don't know enough about it.
Thank you very much for your answer.
Okay, thank you very much. If you have no more question, oh, do you have question, Dr. Kang?
That's my thank you.
Okay.
Logo. Thanks very much today.
Thank you.
Okay, so we are closing today's lecture by Professor Erez Braun. Thank you, Professor Braun, and thank you all, your audience. We see you next Tuesday lecture by Yomo and Shanghai University. Okay.
Okay, thank you very much.
Thanks very much.
Okay, have a good night.
Okay, bye-bye.
Bye-bye. Thank you. Thank you very much.